Hi, everyone. Good morning. My name is Nili Ostrov. I am a postdoctoral fellow with George here at HMS, and I'm here to tell you today about our efforts in the Technology and Infrastructure Working Group. We've been meeting for twice a month for the last year or so, and let me start by what we didn't do. So we did not discuss how to find one way and one technology to build one cell line that was discussed today. Our job was actually the opposite of that. So what we want to do is open an inclusive discussion of different technologies that are available and different limitations of technologies that are not yet available to try to build any of those kinds of cell lines. So here's how we think about it. Our group has industry people, academics, people from different model organisms, and citizen scientists. And the way we think about it is the goal of this project is to build genomes and test them with better technologies and reduced cost. And to do that, we think of 4 major groups of technologies. And even though we chose to do that discussion in 4 groups, that doesn't mean it's the only way to do it, and those 4 groups of technologies speak to each other in many ways. And so before I go into any of those 4, and 10 minutes is definitely not enough to talk about all technologies, I want to give an example of how this group thought about these different technologies and what we came up with for limiting factors and technologies we should be looking at towards this kind of cell line. And so before I do that, if you ask anyone, any scientist or any company, what is the limiting factor? What do you think of these different things in technology is the limiting factor to build this kind of cell line? It depends who you ask. You're going to get very different answers. So we're going to ask you, if you can, during this conference and after, go to the GP Wright website and take a survey. And the survey basically goes through different technologies, and whichever project you do, whether editing or synthesizing DNA or any other project you do, it doesn't need to be a whole genome. Take our survey and tell us what you think is the limiting factor and what technologies do you think we should be looking at to build a genome. But let me give you an example of what we thought of in our discussions. And so the way I'm going to do this is we thought of a technology roadmap as what can we do now, what we cannot do now, what do we need to do short term, and what kind of things we want to see very, very long term. And so we have, for each of the 4 technology groups I'm going to talk about, we have the white area where we can do things right now to improve our current technologies towards a genome, a new genome, a synthetic genome. The yellow is projects in the short term where we need more collaborations, more development to get there. And then red is kind of what we want to see in the future. So the first group is design. When I say design, I mean computational design of a genome. And I think everyone would agree that theoretically we can design a genome, any genome, on a computer, but is that going to be viable? Is that going to work? And so things we can do today, we can very efficiently design small changes. We can design pathways. We can design circuits that we rationally design them, put them on the computer, and then we have rules to make them work in vivo. What's more difficult to do is to design a whole chromosome. So we can have a whole chromosome sequence on the computer. We don't know if the changes we're going to make on the computer are going to be viable. And so things like multi-gene design and large pathway and chromosome structure are things that we do not yet know how to do. Collaborative design is something that not a lot of groups do. I know SC 2.0 have software in-house, and they actually manage their sequences in a collaborative way. But that's something that we will need if we're going to do a whole genome between many groups. In the far future, what you want to see is you want to log in to your favorite organism, and today you have a database that usually gives you names of genes, structure of chromosomes in a very kind of low level. It may be giving you pathways, but does it give you all of the effect of all gene changes? and all metabolomics and all kinds of RNA transcription sequences. No, it doesn't give you all these. So combination of a lot of different databases and collaborative design to eventually have basically whole cell predictions is something we would like to see in the future. Then we go to synthesis. So of all the topics we talk about, DNA synthesis is actually very advanced. It's a commercial available technology. We can all buy DNA, but what are the limitations of that kind of technology when we want to make a whole genome? So we can make oligos. We can build these oligos into bigger genes, and we can probably put those genes into pathways. Is that enough to build a whole chromosome? We can definitely not build an entire chromosome by direct synthesis, one long strand of DNA. But do we need to? Do we really need to have direct synthesis to build a whole chromosome? Probably not. So development of other methods of synthesis, enzymatic synthesis, is something we can look into to reduce the cost of synthesis. There are also things we can do right now within companies and within academia to see how to lower the cost of current phosphoramidite chemistry. But in the long term, we want to look at a combination of those methods to really reduce the cost of synthesis so that it is no longer a limiting factor for building a chromosome. Editing. Editing is everyone's favorite method. Everyone uses editing, and so in a way, editing is the easiest way to go, at least right now. If you ask most people how to change a chromosome, they won't tell you build it from pieces of DNA. They will tell you let's change an existing chromosome to see how we can get to a result. So I think we all agree that a big human chromosome is going to be a combination of efforts from different technologies. And current editing technologies are very good. We have a way that we didn't have before to use CRISPR and Z fingers and TALENs to make very specific changes. But we cannot, at the moment, make 200,000 changes to one cell for many reasons. For example, can you get 200,000 guides for 200,000 different positions into a single cell? No, you can't right now. That's not something that we have done as a community. And so you can have a lot of current usage of the technologies we have. For example, if you had available for the community all the guides for all the position you would like to change, let's say all SNPs, all disease SNPs, if you generated all of those guides and made them available to the entire community, that is something you can do to use current technologies and still get closer to our target. But really for editing, what we want to do is to be able to make those very large amount of changes in a single cell in one big chromosome. And so in mammalian cells, that is still yet to come. We cannot do that yet. And then doing those things requires multiplexing. So what I mean by that, even if we do one change at a time, that is not a feasible way to change 200,000 positions. And so I can give you an example from our E. coli recoding project. There are two different strains we're building. One of them has 300 changes. 300 changes you can definitely do with editing, different types of editing. We use MAGE. You can probably also use CRISPR or TALENs for that. On the other hand, we're trying to make a very, very modified, highly recoded strain with more than 60,000 changes. That cannot be done at the moment with efficient editing. Even though we could do that, that would take almost as much synthesis of DNA and almost as much time and effort to achieve. So instead, we're building that from DNA, by DNA synthesis, de novo synthesis. And so for E. coli, that's enough. We can do that. We can use a synthesis, the synthesis effort to build 4 million bases, but I don't think we can, at the moment, put the time and effort to build from scratch the same way we build an E. coli by just synthesis in terms of cost, time, and labor, the entire human genome, or a recoded human genome. Even just the recoded, even just the coding area is order of magnitude bigger than what we can do right now. And so for editing, there's going to be a combination of current methods and developing abilities to push the editing technologies to higher scale and multiplex. And then finally, assembly and delivery. And so what I mean by that is, let's assume you would make a lot of small pieces of DNA and you want to put them all together in your target cell to make a chromosome. How do you do that? And I think this is where we have the least available methods community-wide to do this efficiently. So we have very good hosts, Cerevisia, to do DNA assembly. Everyone currently that want to do very, very large DNA assembly, they would do that in Cerevisia. Why? Because it has very good homologous recombination. Most other model organisms and non-model organisms that we have don't have that feature. So you can't transform DNA into a mammalian cell and expect it to all come together beautifully like it does in yeast by recombination. So that means that for a lot of the processes to build a chromosome, we would have to use other organisms as intermediaries. So right now we use bacteria and we use yeast on our way to move those chromosomes to other target cells. But even the technologies we have right now, artificial chromosomes are used to, they're like a big plasmid that could hold a lot of DNA, and then you want to move that into your target cell. But those, for example, don't have a human centromere. We just heard from Jeff, remarkable combination of chromosomes to make one big chromosome. That depends on centromere biology. We are not that good at as good as prokaryote in engineering centromeres for human. We still don't have a good way of building a big chromosome in yeast and moving that to human and have it segregate as we expect or rationally design that. And so, for example, centromere biology and chromosome positioning and chromosome structure and folding, those are things that we can start studying right now by forward and reverse engineering to try to understand how to build a synthetic chromosome that will work in the target cell. And so the research can be done now, and eventually we will get to hopefully ways to also do things not just in intermediary, but also in the target chromosome. And so specialized hosts, not just yeast, could be developed with abilities that yeast don't have. So for example, post-translational modifications and specific histones are things that a lot of the single cells don't have and you would like to have in a human chromosome. So you would have to have intermediary host or directly in the human to build in those modifications. And eventually we would like to get, ideally, to rapid building and testing of full chromosomes in their target host. So one option right now is to build a full chromosome in an intermediary host and move it. But eventually, ideally, we would be able to print chromosomes or assemble all of them at once or synthesize the entire chromosome in the target host. So right now we cannot do that, but this is where we would like to go. We would like to be able to control the time, the induction, and the position, and the time of expression of genes and regulatory elements. All these things we want to put together in order to be able to be better at construction and testing of these chromosomes. And so the group has been we have people from different nationalities and different organisms, and what we tried to do is, rather than go through one technology to build one cell line, we tried to discuss different ways of doing this in different organisms. And I think keeping the discussion open for multiple model organisms and different approaches and different expertise by different people will help us, because most technologies are not developed directly in the target host. They require all of us to bring together a lot of different expertise. So with that, these are the active members of the working group, and I remind you to try to chip in and answer our survey, which we will make available to everyone once we have all the results. And I invite Ben and Jeff and Axel and Giovanni to join me.
So we each have, I think, a few comments, but we want to leave time for some discussion. So my name's Jeff Schloss. I'm the moderator of the working group, and I want to touch on a couple of administrative things before I mention another scientific one beyond what I did earlier today. So in addition to the list of active participants, Nili mentioned. We've had actually 65 people express interest in this group. We're very excited about the wide interest, and we're particularly grateful for the people who've been able to take the time out of their busy schedules to help generate the discussions that led to what Nili has presented. As she mentioned, this is a academic and industry, international, and it's been a really informative and some deep scientific discussions. We're very grateful for everyone devoting their time to that. I guess the main other thing I want to say, is that some of the members of our group are actually on other groups, and we've also had side conversations by email or phone with several of the other working groups. So we're pleased to be able to participate in the cross-organizational activities. And finally, I just want to, in addition to the one scientific point I made already about the challenges of trying to engineer genomes when you don't have full accurate sequence of the entire genome yet, I just want to say that one of the reasons I'm finding this project so exciting is the opportunity to iterate for some of the things that Nili and others have talked about. We really need fairly deep understanding of the biology, so for it to be able to figure out how to go about the designs. And that's an understanding of biology that we don't fully have yet. But on the other hand, the development and application of some of these methods are going to tremendously improve our understandings of the biology. And this will go back and forth over the 10 years and probably more future years of the project. So I just find that really exciting.
Then I'll go next. What I really find exciting about the working group is, as Jeff just mentioned, the very broad discussions, and that we also, as Nili described, we're not so focused on one specific approach to get to goal, but really more about taking a very broad look at the technologies. So I'm from industry, and so I just want to make a comment that when you look at the basis of synthesis and whether you produce an oligonucleotide for genome editing, or whether you produce an oligonucleotide to then assemble it into a double-stranded piece of DNA, all of the technology that's actually being used at the moment is more than 30 years old for that. And when you look into ongoing development into those technologies, at least from my perspective at the moment of it, a lot of that is largely industry-driven, which then, because of the nature of industry, often leads to more short-term, immediate payback activities, which in my view doesn't really get us to the goal of hitting the 1,000x reduction in cost, which we need. And so I would really invite the academic community to again re-engage in many of these technology developments. And I think that with the projects that were announced today and a lot of those things, I think gives us really that fundamental research back into this synthesis technology, which I find great.
Mic check. Hello, tech. Oh, we go. My name is Ben Gordon, and I'd like to start by saying what an honor it's been to be acting on this group and having discussions with all these scientists, including all of you in the room that I've only met on the phone. So I hope we meet in person soon. I want to make two quick comments. The first one is, speaking for myself, I think one of the big surprises of working with this group was that the DNA synthesis piece is just one of the major technology obstacles going forward. And I think Nili did a great job of summarizing the four major areas that we kind of broke it down into. But really, new tech on all four of those areas is going to be critical. My main interest is on the design side, and I thought it might be illustrative to tell you how we were thinking about this, and it applies to all four areas. But we're thinking about actual use cases. We didn't have the benefit of seeing what the specification is that Jeff showed this morning. We were asked to think, as Nili described in the abstract. So we thought about what are things that we could do today versus in 5 years. On the design side, that means taking a specification, like you saw on the screen this morning, and reducing it to a functional genome sequence. at least in a computer, which could then be made through all the other steps of the technology. If that genome sequence is a matter of simple changes, most of that technology exists today. But we also like to think about the more abstract designs that we'd like to be able to be capable of in the future, like designing a cell that's a perfect model for a particular kind of disease so that we can go and use it for drug development. Right now, the tools for doing those kinds of things do not exist. And there's several different areas where more work is needed. One of them is just on the, what I'd call the software side, scaling up, instead of dealing with genomes on the scale of millions of bases, genomes on the scale of billions of bases. Another area is in terms of how we scale up the collaboration. So because we expect many, many groups to be involved, the current tools from the SC 2.0 effort are definitely a model for going forward, but they'll need to be adapted. And the third area is, which you just referred to here, which is the biology. We do not yet have sufficiently sophisticated models to be able to predict how huge changes in the genome are going to function and if they're going to function the way that we want. So we're very hopeful that the efforts through the GP Right program will develop those underlying technologies as well as the eventual outputs.
So I'm Giovanni Stracquadanio. I've been working with Jeff and SC 2.0 on designing the yeast genome. So I'm particularly interested in design technologies, and all it goes under the huge umbrella, I think, of computer-aided design. I agree with all my colleagues on this, that we have lots of challenges, but one that I'm really interested in and I think is going to be crucial for the success of the project is about testing. So when you go and write a program, a piece of code, you gotta have some sort of bulb that shows up when says, oh, you misspelled your variable or you made an error that will prevent your code to compile. So right now, at least for SC 2.0, our spell checker, syntax checker was either Jeff or Leslie. Don't hide in the back. I don't think this is going to scale up to mammalian cells. We need to help biologists make decisions. And I totally understand. I come from a mathematics background, and I want to learn the mechanics behind, but I think we have a lot of data that we can use to somehow predict in a marginal way how our design is going to behave. To some extent, I'm talking about all the functional genomics projects and everything. How we are going to integrate into our design workflow, I think, is going to be really challenging. And how we want to make sure that provides reasonable predictions. Because you tune the parameters of your machine learning methods and it spits out completely different results. And the other thing I'm really interested is try to come up with a strategy that allow us to design a chromosome regardless of the underlying assembly technology. I think this is very important. That we have been discussing a bit that somehow there are people that wants to inform the design based on the current assembly technologies. For example, you cannot synthesize long stretches of A's and things like this. I'm more towards an optimistic approach where I think, okay, let's come up with a sequence that we like, and then we will sort it out in 10, 15 years, some way how to synthesize that, or even less, I would say. And this will require what I call now computer-aided manufacturing algorithms. How we can put together this long stretches of DNA in a very efficient way that minimizes our labor and so on, so, which I think are all quite challenging problems.
Yeah, so I wanted to convey the type of discussions that we had mostly, because not just the content of the discussions, which was super interesting, was important here, but also the interaction, which I find is the essence of doing science, is where you get to speak to all these different people that do very, very different things from you, but all try to towards the same goal. And I think the way I know this discussion was good because we got George jealous. I told George on a random conversation what the group was talking about. It's very hard to get George to be jealous of a different meeting. He has a lot of meetings, but he was very excited that we're having this meeting. It's a meeting that, for example, we had industry and we had academics, and we talk about, so how do you get synthesis to be better? And then you have companies that make DNA say, I don't have a market for making one big chromosome, one large molecule of one large chromosome. That's a technology that needs to be developed, but we go by market. So that's an interesting input that you get from, that you don't think about as an academic. Then we had a conversation about cell fusion. And you know, cell fusion is something that we know we can do, but is not yet a routine technology. So we have, you know, the Boca lab, we had Leslie, and we have Bogomil, and we had, you know, Oshimura lab in Japan. We have people doing cell fusion, and all of them tweak it differently, and it's a very early stage of doing that kind of technology, of moving chromosomes from cell to cell, whole chromosomes. And having all these people in one room talking about how we can move a chromosome and which cell can be used to move any chromosome, I think that's the essence of the discussion. And this kind of discussion of whether the design software should or should not include any input from actual technology in the lab. If I cannot assemble more than 1 megabase, should I cut all my design pieces to 1 megabase? Probably not, because I hope to be able to do 100 megabase at some point, so the design should support that. So I think the type of discussions that we had and the type of interaction that you need for this kind of project is to bring together people from very, very different points of view and expertise to try to kind of solve these problems and even think how to think about those. What do you think is the limiting factor? Anyone?
Well, I don't have a comment on necessarily the limiting factor, but you brought it up briefly, the importance of chromatin structure. You talked about post-translational histone modifications, so I was wondering if someone can expand on how you're thinking about attacking post-translational histone modifications, DNA methylation, and chromatin structure with epigenetic editors or something else.
So I'll give you the easy, fast answer. We're going to recruit people that are really good at doing that to contribute their knowledge to tell us what the best way to do that is. That's first. The other thing is that we already have some technologies where specific enzymes are overexpressed in the host. So we already have human histones and centromere-like structures that you can make in yeast, for just a recent example, to try to modify your synthetic chromosome to be more humanized. So there are a lot of different ways to do that. I think in terms of human chromosome biology, there is a lot to do. So one thing we can do is ask human or centromere or chromosome positioning experts, what is the technology that you need to make these better? It could be microscopy. There's a lot of microscopy, but maybe the bringing together of different microscopy methods and maybe funding into these technologies to find out what is the chromosome structure and how to study that would be the smartest way to go. Again, there's no one answer. I think what we want to do is point out, ask the experts in each of those fields, and point out what is the technology that they think we should put money and effort and time into.
We've discussed a lot. We haven't spent a lot of our time, but we've touched on several times the challenge that the technologies needed for this project are just vast, as you've identified a set of them. And I think that's going to be one of the big challenges for the project, is to be able to say, where are we going to try to focus versus what things are we simply, for practical reasons, going to have to try to import from the rest of the community. Fortunately, we're not alone. This project's not alone in trying to develop this whole array of technologies for engineering and testing of genomes. So we're going to have to try to focus without excluding things that we really do need. I don't know how we're going to manage that. Jeff?
So I have a comment on that. So I actually would answer the question in a really different way. While it's true that there's the potential to do chromatin engineering as a really interesting research area, I think for the project we just discussed, and certainly for the SC 2.0 project, as someone who worked on chromatin for many years, this was sort of an obvious concern. But what our work has shown, and the work of many others has shown, both in yeast and in mammalian systems, is that if you put the DNA back as naked DNA, by the time the cells have grown up, they've acquired the right post-translational modification. They've acquired silent chromatin if they're in a part of the chromosome that's supposed to be silenced. I'm talking about yeast right now. More impressively than this, and this is not work from our group at all, but from Doug Higgs' group at Oxford, he showed that you can take a 100 kilobase human alpha globin gene, put it into a mouse embryonic stem cell, and turn it into a mouse, and that mouse will produce alpha globin in the right cells and only in the right cells, using from which you can infer that the chromatin must be right. And perhaps even more impressive set of studies done by Duncan Odom and his colleagues showed that you can put an entire human chromosome 21 into a mouse, and all of the genes on that chromosome show the right kind of tissue-specific expression. So again, hard to imagine that happening if the chromatin wasn't, if not perfect, very, very close to perfect. So in terms of the kind of genome writing that we're talking about for the pilot project, I don't worry about this anymore because of those results.
I want to echo what Jeff said, also having worked in this field for a long time. But there are a couple of large nationally funded efforts in that area. One is the 4D nucleome and one is ENCODE. I don't know if you've drilled down into whether those are useful or not. Some of us have our opinions on that, but they have been massively funded.
The 40 nucleome has been, and ENCODE have been heavily, a lot of the focus has been on population averages, and I think we need more single cell. There is some more emphasis, growing interest on single cell, and I think that will be what we can look forward to.
The data is public, I think, for both of those, but I think it does suffer from sort of there are no standards in it. Cell types are not standardized like you offer, so I think personally I think it's problematic, but it is there, so.
I think, yeah, if there's a question, go ahead.
So this working group has the biggest problem with IP, and how do you think that's going to impact the development of these technologies?
I mean, maybe I can answer it quickly from an industry perspective. I think that when you talk about the IP that's required, I would say that it's really in that pre-competitive state. And so I'm not concerned at all at this stage. And I think, again, from my perspective, even from a large corporation, I think we shouldn't worry too much about IP in early stages. We should focus on the technology and get things done and then sort it out afterwards. Because I think doing it upfront often really just slows things down very, very much. And I think, the approach Jeff described for the SC 2.0 project just be clear about which areas are open source and which areas people can deal with themselves. Things like that need to be put in place, but I think that should be enough.